Hi, good morning. Thank you very much for letting me come here to interview you. Could you introduce yourself, please?
Yeah, so my name is Joachim Goedhart and I'm Assistant Professor. I work at the University of Amsterdam in the Department of Molecular Cytology.
And could you give me a short history of how you actually got here?
Yeah, so I did a PhD at Wageningen University, so there I worked on plant cells. And then there was an opportunity to do a postdoc at the University of Amsterdam, so that was a really nice opportunity. So I went here, and ever since I have been here, so from a postdoc I became an assistant professor. And that's already more than 10 years ago. So
Well, wonderful.
yeah,
It's a really nice institute, as I can see. It's new, isn't it?
yeah. So this building is pretty new.
Yeah.
I think maybe 5 years old. So before we were also in another location.
Right.
So we did a little bit of moving. So
Yeah.
yeah.
It's always nice to be in a new building, I think.
Absolutely. Yeah.
So tell me more about your research. What are you actually doing in
Yeah.
this place?
So what we are doing is we develop fluorescent proteins. Well, they are basically already there, but we try to engineer them, make them better, make them brighter. So that's one aspect what we do, because in the end we want to study cellular processes. So we want to understand how cells grow, how they migrate, all these kind of processes.
Mm-hmm.
And cells by themselves are transparent, so you won't be able to see much in those cells. So we need something to visualize them.
Mm-hmm.
And actually fluorescence is a very nice approach to visualize all kind of processes in living cells. And fluorescent proteins give us the opportunity to visualize all kind of proteins or structures within the cells. So in order to understand those cells, we have to visualize them. To visualize them, we need the probes. So an important aspect of our work is to improve those probes.
Okay, tell me a little bit more about the history of fluorescent proteins.
Yeah, so already in 1960 there was a guy making fishing trips because he found within the sea glowing jellyfish. He harvested those jellyfish from the sea, and he was able to show that those jellyfish make a protein that glows. So then it took another 30 years before the gene that encodes the protein was identified. So all proteins are encoded by DNA, and it was a humongous task to find the piece of DNA that encoded that glowing protein. So finally they were able to identify that DNA. And what was really spectacular is that when they put this piece of DNA into a bacterium or in a worm, then the bacterium or the worm by itself started to glow. So the only thing that was really necessary to make those glowing creatures is this piece of DNA. And I mean, this triggered the whole GFP revolution. So nowadays it's a standard tool in any cell biology lab.
So I understand that you actually make constructs of different colors that are variations of the original green fluorescent protein GFP. How are your constructs different from what was originally cloned from the jellyfish?
Yeah. So the really cool thing is that now you have this piece of DNA that encodes the protein, and DNA you can manipulate, you can change it. It's basically a code that you can change. And if you change this code, then also the properties of the fluorescent protein will change. So you can make fluorescent proteins with different colors, or fluorescent proteins that you can switch on and off. And one of the first things that they needed to do was, because the jellyfish swims in the Pacific, which is pretty cold. So if you put this piece of DNA now into a cell that grows at 37 degrees, you wouldn't see much. So the first thing that they basically did is to change the code in such a way that the protein also folds well at 37 degrees. So that was one of the first steps. So basically all fluorescent proteins that we are using nowadays have those folding mutations. And then a couple of the fluorescent proteins that we made are color variants, right? So GFP glows green. We made some variants that glow yellow, or they glow in cyan colors, or even red nowadays.
That's absolutely amazing. So how are you using these fluorescent bacteria or these fluorescent plasmids for the research that you do? So could you please tell us a little bit about what you're doing, in general, and how do you actually use these different colors in your research?
So one aspect is to engineer new fluorescent proteins. So we basically make libraries of DNA with all different kind of variants. Then those bacteria grow on a plate, and every colony has a unique fluorescent protein, and we try to find the most bright fluorescent protein. So we pick those colonies, and then we start to characterize those fluorescent proteins. And once we have an optimal fluorescent protein, what we then do next is to attach the piece of DNA to a piece of DNA that encodes for a protein that we are interested in. And this fusion protein, as we call it, we can introduce into cells that we're interested in. So those are mainly cancer cells or cells from blood vessels. We grow them on a dish and we introduce this new piece of DNA into those cells. And that allows us to visualize the processes that we are interested in. So then we follow those cells with a microscope, do something that we call lifestyle imaging, and track those cellular processes over time.
And so how do you use different colors at once? What do you use the two different colors, or three, whatever, how many you use at once? How do you do that? What do you do with so many at once?
Yeah, so we want to visualize multiple processes over time, so we can make correlations to see how two processes affect each other. And when you have different colors, with our microscopy setups, we can separate those colors. So you have specific filters that only pass, for instance, the cyan light, or only pass the yellow light. So you can easily visualize up to 3 different colors, but there have been efforts to go to 6 or even 10 different colors. And the more colors you have, the more processes you can visualize at the same time.
Okay, thank you. So this obviously, you're taking a natural gene out of an organism that is the jellyfish, and then you're making changes to it, and then you're putting it in different systems. That sounds very genetically modifying. And I know in the public eye, GMOs, genetically modified organisms, are very, it's scary for the public because they think that it's something unnatural. Could you comment a little bit on that?
Yeah, so indeed this is manipulation that we do with DNA. So we also introduce this piece of DNA into an organism, so it's a genetically modified organism. And this effort is also something that's called synthetic biology, because you take different parts from nature that you assemble, and this enables you to make some complete new functionality within cells. So you could also use this, for instance, to take different parts from nature, assemble this into a cell, and in this way introduce a new metabolic pathway, for instance, that makes biofuels. So it also gives us endless opportunities to create new components, new materials. Yeah.
Thank you very much. All right, well, I think it's time to see how it actually is done.
So what I will be doing, I have a culture of bacteria over here. They are at this moment non-fluorescent, so they don't produce the protein. And what I will do is I will stream the bacteria onto the plate. The plate contains a sugar that will induce protein production, and then the bacteria will grow, produce the protein, and the next day we'll have a fluorescent plate.
Great, great. Looking forward to seeing you do it.
So let's see how it looks after incubation.
And remind us again, this is a 37-degree incubator?
Yeah, okay. It is.
Yeah. Wow, look at that. That is wonderful. You can already actually see the fluorescence under normal light. Brilliant. Okay, so tell us where we are now. What are we doing?
So here we have a UV tray. So it's kind of a black light. So it happens when you turn on the black light, it will excite the fluorescent molecules, and they will release their energy by emitting light of a different color. So the black light is UV, you won't see much of it, but the colors that are emitted are visible light, so those you will be able to see.
Okay.
So let me turn on the black light.
Wow,
and
look
you
at
can
that.
nicely see the glowing colors.
That is fantastic. So let's turn off the light, and it becomes even more amazing. Look at that. Fabulous. And how long will this last for, this fluorescence? If you put it like this for, how long will it actually glow this brightly?
Yeah, it will depend whether you switch on this black light. So if you don't switch on the black light, the fluorescence will, or the protein will be there for a very long time.
Mm-hmm.
But when you excite it, it will also be partially destroyed. I'm not sure how long it will last on the UV tray, but it will slowly fade away.
Okay. Well, it looks absolutely amazing at the moment. All right, so tell us where we are.
So here we are in the microscopy room where we take the images of our cells. And this is a typical microscope image that we have acquired, in which we use the fluorescent proteins to visualize structures within cells. So there are actually 3 cells, and we have taken different fluorescent proteins to either tag the Golgi apparatus, the nucleus, and the mitochondria, the powerhouses of the cell. And each individual image is taken with a different fluorescent protein. That's why we can nicely separate them and visualize them independently. And after acquiring those images, we can make an overlay so that you see over here in which different fluorescent proteins are shown in different colors. And you can also see how those compartments localize with respect to each other. So for instance, we have here the nucleus, then the Golgi apparatus next to the nucleus, and the mitochondria are distributed all over the cell. So this is the typical data that we acquire with those fluorescent proteins.
So what exactly was the experiment here? What were you looking at typically?
So in this experiment, what we try to do is optimize the localization of those fluorescent protein fusions. So there are a lot of these kind of constructs to visualize structures, but they usually are a little bit fussy or don't localize exactly to the right structure. But for instance, here with those mitochondria, you really get a very nice crisp image of those mitochondria. So this was just to show that this is a beautiful plasmid to visualize mitochondrial structures.
Why isn't that cell on the corner? Why doesn't it actually have the mitochondria? Did that not take up the plasmid, I guess?
Yeah, so we do a transfection with plasmids, and this uptake of plasmids is it's a random process. So some of the cells get more of the plasmid than others. That's indeed why you see this cell shows less of the mitochondrial fluorescence. Yeah.
Wonderful. Thank you very, very much. So this is really, really interesting, Joachim. Thank you so much for letting us come in and record you in your lab. I mean, these colors are absolutely fascinating. Is there anything else that you would like to tell our audience today?
Happy Christmas.
Happy Christmas. That's a great message. Thank you very much for having us, and hope to maybe be back soon when your research is a little further down the line.
Yeah,
Thank
sure.
you.